gastrointestinal system and the urinary system. بكتي بس يو systemana again بكاتتاني chona histologically what is the difference between these three systems. Regarding the first one, which is the respiratory system, as you know, this system has a tube-like structure and it ends in the both lungs. So we have anatomically two portions, conducting portion and the respiratory portion. The conducting portion, from its name, is that part of the respiratory tube or the respiratory system that conveys the air from outside environment to the lung and to the rest of the body. While the respiratory portion, again from its name, it is that part of the respiratory system that is responsible for the respiration process or the exchange of gases. Regarding the conducting portion, anatomically it consists of the nasal cavities, starts from the nasal cavity, nasopharynx, larynx, trachea, bronchi, bronchioles, and lastly the terminal bronchiole. So the terminal bronchiole would be the last part of the conducting portion. After that, the respiratory portion starts. It's where the gas exchange occurs, as we discussed, and it's consisting of three parts. The respiratory bronchiole later on opens to alveolar ducts, and the alveolar ducts open to the alveolar sacs or alveoli. So if we look at this diagram, we have both the anatomical and the histological classification of the respiratory system. Regarding anatomical classification, we have two parts, the upper respiratory tract and the lower respiratory tract. That's how we divide the respiratory system anatomically. Regarding the upper respiratory tract, it starts from the nasal cavity and the related frontal and sphenoidal sinuses, regarding also part of this section of the respiratory system, and the respiratory pharynx, okay, till the larynx. Here, till the larynx. While the lower respiratory tract starts after the larynx and distributes to the trachea, which opens to the main right and left main bronchi, and this will divide later on to bronchioles and terminal bronchioles, and later on will join the first part of the respiratory portion, which is the respiratory bronchiole. Regarding the conducting portion, we give an example on it is the trachea. The characteristic epithelial lining of the trachea, which is part of the conducting portion of the respiratory tract, is the characteristic respiratory epithelium, or we can say the epithelium is the ciliated pseudostratified columnar epithelium that we studied in the epithelial session. So if you look at this slide on the left side, it is stained with a special stain. You can see red color and blue color, which is not usual. Okay. Regarding the type of the epithelium which is present in the trachea or the rest of the conducting portion of the respiratory tract is the ciliated pseudostratified columnar epithelium. We mentioned that in the lectures of the epithelial tissue. We can see here on the left side the slide which is stained with a special stain that is not the routine stain of H&E. Here we can see the cavity of the organ or the surface lining of the trachea, and this is the epithelium here, starts from here to here, reddish, somewhat reddish in color, and you can see the cilia on the surface of the trachea. And below the epithelium, this is the full thickness of the epithelium. You can see between them scattered the columnar, pseudostratified columnar epithelium with the goblet cell. So this G letter here refers to the goblet cell, while the C stands for the cilia, and the BM stands for the basement membrane or the basal lamina, which stains here blue in color. While this region here, below the basement membrane, is the full thickness of the lamina propria. As we mentioned, lamina propria is part of the connective tissue. It is the loose areolar type of connective tissue which underlies the epithelium of these tracts. For example, the respiratory GI tract or the urinary system. While on the right side, you can see an electron microscope photo for the surface epithelium lining the trachea, which is the characteristic respiratory epithelium. You can see the large number of cilia. In between those cells, who are the goblet cells, they don't have cilia. So these scatter between the ciliated columnar cells. Regarding the constituents of the respiratory epithelium, you have these 5 types of cells. First, the ciliated columnar cells, as you know, which is the usual part of the respiratory epithelium. It's about 30% or somewhat 50% of the rest of the cells of the respiratory epithelium. And later on comes the goblet cell, which is somewhat lesser in number, and brush cells. And we have the basal short cells and the small granule cells. So as you see here, different types of the cells found in this type characteristic epithelium of the respiratory system or the trachea, for example. Here on this slide, you can see the cilia is clear. Goblet cells, which are large, flask-shaped cells filled with mucus secretions, so they appear faint in color or clear in color under the microscope. In between, scattered the different levels nuclei of the pseudostratified epithelium. Regarding the trachea, if we discuss it as a tube-like structure, to study it histologically, you have to section it. The best way is to section this tube structure transversely, and you start to count the layers from inside to outside. This is our usual way to study the layers in histology, specifically in hollow organs or the cavities or tube-like structures. So the trachea has many blood vessels found in the region of the lamina propria, which is the region of the connective tissue underlying the epithelium. This has role for warming the air beside nutrient supply. And we have numerous seromucous glands. Those glands are distributed in the region of the submucosa, as I will show you. And we have, of course, the goblet cells. The goblet cells lie within the epithelium itself. So beware in the examination if you ask about the position of the goblet cells. Is it in the epithelium or in the lamina propria of the connective tissue? You have to say that it is in part of the epithelial tissue in the superlying the connective tissue. And we have also 16 to 20 C-shaped rings of hyaline cartilage. I think you've been studied this in biology sessions before. This keeps the tracheal lumen open, and we have lymphoid system cells that lies just under the epithelium as part of the lamina propria. So here, in this slide, this is a histological slide which is stained by the routine H&E stain. You have the tracheal lumen. Here, this is the tracheal lumen. This is the cavity of the tube. We have a part or a C-shaped part of the wall of the trachea. So you'll start counting the layers inside and outside. The first layer would be the respiratory epithelium. Here the slide is with low power, so you can see from far away, you cannot distinguish which type of epithelium. It is unusual in the exam to put a slide with low power and ask you what type of epithelium is this. Okay, it should be always with high power or mid power. So here, the respiratory epithelium, we know as trachea it is this pseudostratified columnar epithelium, ciliated epithelium. Beneath it, we have a thin layer, which is, this is usually dark in color, purple in color. Beneath the epithelium lies the thin layer of the connective tissue that supports it, which is usually the lamina propria or the loose areolar type of connective tissue. This one is rich in the lymphoid tissue cells and lymphoid nodules in the three tracts, in the respiratory and in the digestive, also in the urinary, found those lymphoid nodules in the region of the lamina propria. Beneath the lamina propria, after this thin layer of connective tissue, we have a thicker layer of connective tissue, which is not loose areolar, it is the dense, irregular type of connective tissue. This forms the submucosa. Okay? So it's a lamina propria, it is the loose areolar type of connective tissue. While the submucosa, the layer behind it, is the layer of dense, irregular connective tissue type. Here in this region of the submucosa, this is much thicker can see a section of glands. Okay, secretory portions of glands. Those are the seromucous glands of the tracheal wall. Usually they are found in submucosa. أو لو كشنا نزور زور بين كأبي مثلا الامتحان يتعلم أبرسي. seromucous gland لو كشنا كده electroid trachea. يعني أولي بس بين trachea. Where exactly in the trachea? After that, we have the region of the cartilage. So this violet color structure here, thick layer, is the hyaline cartilage. And the next lecture of the bone and cartilage will discuss how you can distinguish it under the microscope. If you compare it with the submucosa, it is much violet or much intense bluish in color than the layer which is the dense irregular connective tissue or the submucosa, which is somewhat faint or pinkish in color. So these are the layers. If we count them, mucosa starting from the first layer, and the mucosa is composed of the respiratory epithelium and the lamina propria. Carbon, culling, shape, mucosa bactericanin, it is the epithelium plus lamina propria. Okay? Later on comes the submucosa, as I discussed. عادة السبmucosa تشي هي glandular here. طبعا nerve supply here, blood supply here, بس عادة lamina propria the blood supply is richer than in the submucosa. Duotarchia, the fibrocartilaginous layer, as we mentioned, which is about 16 to 20 C-shaped rings of hyaline cartilage. They keep the lumen of the trachea intact or open. If we take another slide for the as a cross-section for the trachea, this slide is important. You can see this is the surface of the cavity of the trachea. We have this shadowed area. This represents the epithelium. This is with high power slide. So this is the respiratory epithelium, and later on comes this area which is the lamina propria. Later on submucosa, somewhat thicker than the lamina propria and contain these structures. These light structures, they represent the cross-section of the secretory portions of the glands, the seromucous glands. And their color, they are usually very faint in color, or grayish in color, because their content is the cells that produce the mucus that replicates the surface of the trachea. So they appear lighter in color than the surrounding submucosa or connective tissue. Later on comes the bluish color or purple in color tracheal cartilage. That's how you distinguish the layers there by them. Regarding the respiratory portion, so the trachea was an example of the conducting portion. Most of the conducting portion have the same histological layers with little difference. Regarding the respiratory portion, we will take the alveoli themselves, the sacs, where the process of the gas exchange occurs. So alveoli are sac-like vaginations of the respiratory bronchioles, alveolar ducts, and alveolar sacs. Structurally, alveoli resemble small pockets that are open on one side. The structure of alveolar walls is specialized to enhance the fusion between the external and internal environment. And we have what is called alveolar septum, which is a wall between two neighboring alveolar sacs. This septa is vascularized with richest capillary network in the body. And we have three types of cells in the wall of the alveolus. We will discuss these later on the diagram. We have type 1 alveolar cells. They are extremely attenuated or thin cells. They compose about 97% of the whole cells of the alveolar surface. The main role of these cells is to provide a barrier, okay, of minimal thickness that is ready to exchange and permeable to gas. Type 2 alveolar cells, they are round cells that often occur at points of where the alveolar wall unite or meet. It gives rise to the pulmonary surfactant that lowers the surface tension. And lastly, we have the alveolar macrophages. They phagocytose, as you know, erythrocytes, or any foreign bodies or airborne foreign materials that are trapped in the alveolar space. So here in this slide, we have two photos. One is an illustrating diagram for the wall of single alveolus and a histological slide. So here in the diagram, you can see this is the composed, if we count the layers from inside, this is the cavity of the alveolus. You can see here the first layer is composed of the simple squamous type of epithelium. Very thin, attenuated, simple squamous type of epithelium, and usually because the diameter of the alveolus is small, so it has mainly two cells, only two cells, or sometimes one cell of the thin attenuated simple squamous type of epithelium, as we see here. So this is the nucleus of one cell, and this is the nucleus of another cell. Well, this large cell, which is protruded into the lumen of the sac or the cavity of the sac, this is the type 2 pneumocyte, or sometimes called great alveolar cells. They are the same. The great alveolar cell and type 2 pneumocyte are the same. While type 1 pneumocytes are the thin attenuated cells. Okay, so we have, this is one sac, and this is another sac here, another cavity. In between, we have the interalveolar septum. Usually, this interalveolar septum between the two sacs has what? Has a rich capillary network. And in the lumen of the capillary network, we have the lining endothelium. Capillary cuticle, you start counting the layers, has the first layer, which is tunica intima of the capillary. That is represented by a single squamous cell, thin attenuated squamous cell, which is called endothelium. We mentioned that in previous lectures. So here, this is the capillary, this is the lumen of the capillary, and these are the RBCs inside it, and this is the nucleus of the endothelium cells lining the capillary. So it is a very close relationship between the endothelial cells of the capillary and the pneumocyte type 1 cell that will allow the gas to exchange between them. On the left side, you can see the sac here. This is the cavity of the sac. This is the wall of the alveolus, or the wall of the sac. You can see a group of nuclei, attenuated nuclei. This is one nucleus. This is another. This one is another. These are the nuclei of the pneumocyte type 1. which is the main cell. Well, this rounded large cell, which is found at the point where the two sacs meet, this one is the great alveolar type, or great alveolar cell, or alveolar type 2, or sometimes pneumocyte type 2, that produces the pulmonary surfactant factor that reduces the tension in the wall of the alveolus. And we have the macrophages, also the macrophages. The macrophages distribute usually here, near the cells which is lining the wall of the alveolus, or inside it, or inside the cavity. So this was regarding the first inner body surface, which is the respiratory system, both the conducting portion and the respiratory portion. Regarding the second inner body surface is the digestive tract. As we know, it is again a tube-like structure. It starts from the mouth and ends in the anus anatomically. So it has different parts, and it is a long tract. And it has inner body surface. And this inner body surface, as you know, is lined by special type of epithelium that you will mention. And if we take this tract and cut it transversely, we will again start to count the layers inside to outside. So the layers of the wall of the GI tract in general is first the mucosa, which is formed of three sublayers: the lining epithelium, lamina propria beneath it, and the muscularis mucosae. In whole the tract, the lamina propria and the muscularis mucosae is the same, while the epithelium changes from one part to another part. Later on comes the layer of submucosa, the second layer, which is the dense irregular conductive tissue. And number 3 is the muscularis propria, sometimes called muscularis externa. Helena, okay, when you hear externa, you know it is the external layer of muscle. So we have two layers of muscle. The inner one, which is muscularis propria, the inner layer is muscularis mucosae, and the outer layer is muscularis propria or muscularis externa. And lastly, we have the outer part of the tube is the adventitia or serosa. And I will discuss that with you, the difference between adventitia and serosa. So here in this diagram, this is a cross-section through a part of the digestive tract in general. Nalin and stomach, nalin and pharynx, or esophagus in general. We will be counting the layers as it is the same in different parts of the GI tract. So from the inner layer, the first layer, this is the cavity or the lumen of our tube. The first layer, the pinkish one here, it represents the mucosa. Mucosa shukmanchia. Mucosa is further subdivided to 3 layers: the epithelium, lamina propria, and muscularis mucosa, which is the inner muscle layer, the thinner one. Okay? Later on comes the submucosa. This one. This is the submucosa. It is thicker than the mucosa. The submucosa is the dense irregular connective tissue, and most of the time it contains the glands. Okay, as in the case of trachea. We have different glands. We have mucous glands, we have serous glands according to the part of that part of the tube, digestive tube. Again, we have after that the muscularis externa layer. Muscularis externa or muscularis propria is usually of 2 layers, but in the stomach it's of 3 layers. This is the only difference. So in general, it is of inner circular layer and outer longitudinal layer. شلون أسامي circular and longitudinal؟ لقد تعطيك slide okay? Of fiber that running circularly, these are the inner circular part, while those who are cut like dots, as I will show you, these are the longitudinal layer of the muscularis propria. And lastly, we have the serosa. Serosa or adventitia. Serosa adventitia is again a connective tissue layer, which is dense irregular connective tissue layer. So this layer, sometimes, in some parts, it is covered by an extra layer of mesothelial cells or simple squamous cells, which is called mesothelium. In some parts, they are not covered. Mostly it's covered with serosa. So agar hatu gadara layer connective tissue ka layer kisai epithelium بسروبه, a simple squamous layer. This is called serosa. Well, agar نبي simple squamous نبي just the connective tissue itself, so it is called adventitia. طبعا it differs from part to part of the body, and usually the serosa layer or the mesothelium layer that covers the adventitia, it comes in areas that we want to lubricate that organ or to prevent friction between different parts of the tube. For example, the intestinal tube, and it is compact and compressed in a small abdominal cavity. Okay? They will adhere and they will have friction between them, which is damaging to the tissue of the tract. So we need to have this extra layer covering the adventitia, which is called serosa. We have in this diagram both the sections, the transverse and the longitudinal. So this is the transverse one, while this one is the longitudinal section. So you can see the different layers that compose the GI tract. Starting from the lumen, which is the first layer, this is the lumen of the canal. The first layer is the mucosa. The mucosa composed of these 3 parts: epithelium, lamina propria, and muscularis mucosae. Muscularis mucosae is a very thin strip of smooth muscle, comes beneath lamina propria. هذا هو صوت الكلمة. احتمال سلايد احتمال نرجع تذكر فيه لأن مشان ثانية. And later on comes the submucosa here. So this was the epithelium, the mucosa, and the epithelium. Beneath it is the submucosa. Submucosa is usually containing the submucosal glands, blood vessels, and nerve plexuses. For example, here in the longitudinal section, you can see this yellowish network structure. This resembles or represents the submucosal nerve plexus. It is a special nerve plexus, part of the autonomic nervous system, which is found in between the layers of the muscles in the wall of the GI tract. Their function is to send signals to that part of the body for the sake of the peristaltic movements of the GI tract. يعني أجر أمنية plexus أنا معناه نعمل so the food material or the digestive material they will be stuck in the GI tract. They will not move. So these two parts of the nervous system, both the submucosal nerve plexus as here, which is found in the region of the submucosa, where you was submucosal according to its location, and the outer one, which is here, as you see here, the myenteric nerve plexus. This is another part of the anatomic nervous system, and found between the layers of the muscularis externa. Yet can submucosa, the other one is between the two layers of the muscularis externa. Again, they are responsible for the movement of the external muscles. And from outside, we have this layer, which is pinkish here in color. This resembles the serosa, represents the serosa. As we mentioned, the last layer, which is the adventitia, with a layer of simple squamous epithelium over it that make it lubricant and resistant to friction. The functions of the digestive mucosa in general, starting from the epithelium lining, it provides a selectivity permeable barrier between the contents of the tract, the digestive material, and the tissues of the body. Facilitates transport and digestion of food, promotes the absorption of products of the digestion, produces hormones that affect the activity of digestive system, and produce mucus for lubrication and protection. Each cell of this digestive mucosa has its specific function, according to the cell type. Regarding the lamina propria, which comes beneath the epithelium, is a rich zone in macrophages, in lymphatic nodules, lymphocytes. So it's part of the protection system or immune system in the tract. Later on comes the muscularis mucosae, which is the very thin strip of muscle, and usually this one is, of course, involuntary. It is a smooth muscle type, not skeletal muscle type. Later on, the submucosa, which contains the submucosal mesenteric plexus of the autonomic nervous system, and later on the muscularis. We talk about muscularis, we mean muscularis externa. It is composed of smooth muscle cells of two sublayers, inner circular and outer longitudinal. أولا هنتين غلط بين الامتحانات in the connective tissue between the muscle sublayers of the externa, we have the myenteric nerve plexus. Later on, the serosa, which is a thin layer of loose connective tissue rich in blood, vessels, lymphatics, and adipose tissue, and which supports the intestine. By the way, the adventitia in some textbook it referred to it as a dense irregular type, and some reference book as the loose type. It depends on the location. لبعض شينا adventitia dense irregular لبعض part of the tract is the loose one. So it is different. In general, it's a connective tissue type of epithelium. Regarding the esophagus, is part of this muscular tube, which is the GI tract. أجزاء layer كان يشملوا من باسكين. The lining epithelium or the mucosa أجزاء سكين epithelium كين. بشوطنا بس ما كل حظر epithelium وطبعا it's lined by the non-keratinized wet type of stratified squamous epithelium. Okay, so it has no keratin there. It is the wet type or the moist type. And later on is the submucosa layer beneath it. طبعا lamina propria يتهيأ بالتأكيد epithelium lamina propria and muscularis mucosae. Later on comes the submucosa layer, submucosa esophagus. Usually you find the esophageal glands. Those glands they secrete mucus to lubricate the surface. Aguilera series slide on the left side we have this high power slide staying with H&E, where the E represents the full thickness of the epithelium from here to here, thickness epithelium. Okay, this is the lumen of the organ, okay, or of the esophagus, and this is the full thickness of the epithelium. Because it is the stratified type of epithelium. Layer is also there. And you can see here no keratin there in the cell. It is the moist type, and you can even see the nuclei of the live cells at the superficial layer, keratin even, which is a dead layer. Beneath it, we have here the lamina propria, which is pinkish in color. And then the cross section would be whole tube. كاملين أتواني وسيركل بيبيني في الامتحان أبوها برشة ويا بوتبي يعني برشة ويا باشا. So we'll start from the lumen, and this is the epithelium, lamina propria, and if you concentrate here, you can see this thin line. as بقول لك واضح كيف. This thin line, this is representing the muscularis mucosa, which is the thin strip of the smooth muscle. The first smooth muscle, which is very thin. Later on comes the submucosa here, which is somewhat pinkish in color. And while this one, عادة في المصل الجير microscope, رأيك pinky, intense ورقة, sometimes it is reddish in color. باشا. So بقول شوية ناسيتو the muscle layers. And if you see here, we have two orientations. From here to here, we have one orientation. The fibers are running circularly, and here they are cut. So these are the cross section of the longitudinal muscle fibers of the outer layer of the muscularis externa. الثاني الأطلسة كشفي بيني لكتير كان الكتموناردن قالوا كرتانو بتاعي أطلسة رسمي زور جواني تاعي for the layers of the ossifragus. So you can study it. Another slide. Outside. The last layer. Here. Maniera that's a good one. Watch the maniera below power You cannot see this serosa. Why a longitudinal layer? After that comes the layer, which is a very thin layer. And if you magnify it with high power, you can see the thin layer of connective tissue, and above that the layer of the mesothelium, which forms the serosa. Here again, another slide for the lumen of the ossifragus. This is the lumen, this is the epithelium. Epithelium. This intense color violet part of the lumen is the full thickness of the epithelium. And mucosa it's just the epithelium of the mucosa. يعتبر أما lamina propria بس بيها طبعا شكل high power جوهر الدرجة epithelium, lamina propria. And this thin strip of muscles, this one, أمانة reddish in color somewhat. This is the muscularis mucosa. And later on comes the submucosa, submucosa. You will see light circular structures, which are the secretory portions of the ossevagal glands. طبعا الأركان يقدروا يدركوا which is muscularis externa and the serosa. As I said, and according to the power. Power can be low power, but money structure can only be high power, you can see only the lumen and 2 layers after that. Another part of the tract is the stomach. The stomach is a mix, as you know, mixed exocrine and endocrine glands. So it's responsible for the digestion of the carbohydrates, protein, and triglycerides. And by the way, the stomach has 2 types of digestion. Both chemical digestion, which is by the hydrochloric acid and the enzymes that they secrete, the hormones, and the mechanical digestion. Carbohydrates will not be digested in the stomach, it will be digested in the mouth by the salivary glands. Digestion it's a complex process. It starts from the mouth, but it will be completed in the rest of the digestive tract. So the mechanical digestion, the muscular movement of the walls of the stomach helps to digest more carbohydrates. So it's part of the process. About the lining of the stomach, and the stomach is a dilated portion of the tube, but it is still a tube. It is part of the muscular tube of the GI tract. It's dilated, and the muscles of the externa is of 3 layers. they are well developed and consist of 3 layers. هل level away and mechanical contraction of the stomach will be more powerful, so we need an extra layer of muscle. Regarding the epithelial lining, is simple columnar epithelium. أولا لو حاضر epithelium بس ما the stomach is lined by simple columnar epithelium. And it has gastric glands. This is a characteristic feature of the epithelial lining of the stomach. And gastric gland, as I'll show you in the next diagrams and the photos, it has different types of cells. If we start to count them, we have the stem cells. They give rise to the epithelial cells. And we have mucus or neck cells. Mucus and neck cell epithelial. Mucus epithelial, they produce mucus. Neck epithelial because they are found at the neck of the gland. Later on comes the parietal cell that produces the hydrochloric acid. The chief or zymogenic cell that produces pepsinogen. And later on the various enteroendocrine cells that secretes hormones or part of the endocrine system in the GI tract. This is the surface epithelium of the GI tract. Mucosa. This whole thickness is mucosa, composed of the surface epithelium, which is the simple columnar type of epithelium, the geriatriate, which is the loose alveolar connective tissue, duotrichate, a very thin strip of muscularis mucosa. So, mucosa. Beneath the mucosa, we have submucosa. عادة submucosa gland blood nerve supply. Later on, we have muscularis externa, beside the inner circular and longitudinal, we have an extra layer, which is the oblique one. What layer in the stomach? Yes. The layers of the stomach, muscularis externa. Okay. So we have the oblique layer, the circular layer, and the longitudinal layer. 3 layers. مفروض طبعا الجير سلايد شابين نسيتو اگر هاتو سلايد يكتبوها وقوانتر من دانا بين السليرها جيكي لو يتقضي اما نسليرها باشا اگر play اما oblique, longitudinal line and circular. So you have to concentrate on the slides. باشا اگر بين بس gastric gland we will take one gastric gland, which type of gland? بس gland came per se classification of oil بس ما كلها محاضرة كانت بيشوف. Which type of gland? It is simple tubular. Simple of all level away تنهاية ducty here. This is the gland. باشا. هي ducty here. يعني branch ناكا ductata, so it is the simple type. And it is tubular type because the structure of the secretory portion كأنه شيء خواربيتي. Here, this is the secretory portion. This is tube-like. It is not acinar, not sac-like. But again, see the silicon came here from the secretory portion. The way came, which my cell is on. Here you have the mucous neck cells, the here. Here we have the mucous neck cells. The acidophilic type cells, because they are in pinkish in color. These are the parietal cells. Later on, we have deeper here. We have the zymogenic cells. And at the base of the gland, we have the enteroendocrine type of cells. If we look at this slide, which represents to us the full thickness of the mucosa and with its gastric gland, so we have here the lumen of the stomach, and we have the surface epithelium, which is covered by the usual simple columnar epithelium type. And we have the pits, a group of pits, which are openings into the mucosa. And in the end of the pits, at the basal part of the pits, there is the gastric glands with its different type of cells that we mentioned. Stem cell the basal part of the gland, while the mucous neck cell, they are found at the neck or the start of the gland. Some glands are short, some glands are very long. They may occupy the full thickness. يعني بعد زي في أجاتا error. So the full thickness of the mucosa. أجر هاتو أم glandana heavy. These are the glands. What are these thin strips between the glands? What do you think? We have the epithelium, which is the simple columnar epithelium. We have the glands that enters throughout the full thickness of the mucosa, and we have thin strips in between them that separates the gastric glands. Yes, these are the connective tissue, which is the thin layer of lamina propria. Masha? Going on. And what is this layer? This is the muscularis mucosa, which is the thin strip of muscle. Vertebral mucosa. Regarding the small intestine, مجموعة معلومات في شيء ولكن to distinguish it from the stomach and distinguish it from the esophagus. It's composed, as you know, of the duodenum, jejunum, and ileum. One characteristic feature is the presence of villi, and we have the glands of Lavernois, which is the name of the glands in the intestine. So again, what are the intestinal glands? You can name them. They are the glands of Lavernois. Going basi feature can indicate the histological features. And one characteristic feature also of the small intestine is large number of goblet cells, which are found in the lining epithelium, just like in the respiratory tract. And another feature is plica circularis, or Kerpering's valves. They are found, or they are deep permanent holes. They are found in the mucosa or submucosa of the intestine. Here in this slide, you can see these structures, these folds. These called plica circularis. They are spiral folds. They are permanent folds found in the wall of the small intestine. Lechibia catwa, fold like a mucosa or submucosa. While the villi here, or like a finger-like projection, بالمعظم بس مثل حظري epithelium. The villi are finger-like projections. They are formed from the folding of the mucosa epithelium itself, not the submucosa. While plica circularis is permanent, spiral, and composed of mucosa and submucosa. Villi are covered by simple columnar epithelium. The cells of the intestinal glands, a simple columnar epithelium, which is the usual type of epithelial lining of the intestinal tract. We have the glands of the intestinal gland, which are the crypts of Lavernois. Cryptica, gastric gland, they have the absorptive cells, which are the simple columnar cells, goblet cells, palate cells, amanophil cells. They are regarding part of the immune system because they release lysozymes for the digestion. And we have M cells, also M cells, they are part of the immune system in the lining of the intestine. We have stem cells that produce all the four or five types of cells, and we have the enteroendocrine cells or a pod cell. طبعا enteroendocrine cells, they are rare, and they usually produce certain hormones which regulate the process of digestion. باش أقرأ slide سيري كان single villus, gift finger projection, cross section معنقدوة type epithelium مثل sericea is the simple columnar epithelium. باش nuclear pan they located basally, so it is a simple columnar epithelium with the flask-shaped cells or number of goblet cells which are flask-shaped as you see here in this slide and full with mucus, while the nucleus and the other organelles of the cell they are located basally. The large intestine, what is the difference between it and the small intestine? There is no villi. يعني إذا هذا large intestine we will cross section, in operation or for example as an autopsy, you will see the surface epithelium is without villi. It is smooth. Small intestine. We have presence of glands. Again, we have intestinal glands special to the large intestine. We have a marked increase of goblet cell, يعني إجمالي goblet cell هرجكاري زور زيادة cells like كأيبينين. We have two other features, tinea coli and appendices epiplosi. These are, we can say these are anatomical variation rather than histological variation. عشان كباندك مصنع كنت بشيء we can see it with naked eye. شكاني maybe histologically. So this must be an anatomical variation. So regarding the epithelium of the large intestine, again, is simple columnar type of epithelium. باشا goblet cell معناها تجمع كي زورزورة بس stem cell معناها these types of cells they are found in the epithelium. يراو diagram study potential or ETFA. By the way, I'm trust me diagram account or any illustration is not for the examination, it's for your sake, just to understand the next tube histological slide. Verace Avini, this is the surface which is smooth, Basha. يعني Nephlica circularis Avini هنا finger-like projection Avini, which are the villi, smooth surface. But you have the pits. Those pits, they are the opening of the intestinal glands. Basha, this is one gland, this is another gland. طبعا ترتقي شيء large intestine to the small intestine. It is somewhat shorter and more straight. But small intestinal gland or the crypts of Laverton, they are longer and more tortuous. Basha, classification gland again they are simple tubular. Basha, simple tubular. يعني ducty hair, it is tube-like secretory portion. These are the glands. These are the intestinal glands. They are shorter and somewhat more straight than the crypts of Laverton. These are the cells of the gland. كسيري كي أميني ركي زور كاونترا. الجلاند كاني stomach. الجلاند كاني small intestine. Why? Because of large number of goblet cells. Goblet cell is also سيايا. ويقوم الcell كان. The other types of cells will decrease in number on the expense of the goblet cells. ويقاتل ذلك. So on the exam, if you have a slide for the small intestine and large intestine, you have to distinguish them. Depending on those criteria. Regarding the urinary system, which is the last inner body surface that we will discuss, again, it is a tube-like structure. We will not discuss the kidney, it is part of the urinary module. Or inshallah, the upcoming modules. Next year, for next sessions, you will take it as an organ histologically, and I will discuss it to you. That's what the tube occurs. The urinary system consists of pair kidneys and ureter bladder and urethra. This system helps to maintain homeostasis, as you know, has the function of filtration of cellular wastes from the blood, selective reabsorption again of water and solids, and excretion of waste and excess water as urine. We will take this gland, this tube, and we will cut it again as we did in the respiratory and in the digestive tract and start to count the layers. We have the lumen of the muscular tube, for example, the ureter here. It is a muscular tube. The first layer will be the epithelium. Basha, epithelium it is the transitional type of epithelium. So you have to be familiar with it. So this is the characteristic epithelium of the urinary passages, and we have the lamina propria beneath it, which is the loose alveolar connective tissue, and we have the muscularis layer. And outside this thin layer, which is gray in color here, it represents the adventitia. Adventitia, the urinary system again, could be serosa and could be adventitia. Serosa nima. Bladder, urinary bladder, surface urinary bladder is covered with serosa. Loose connective tissue here plus one layer of simple sponge epithelium. Particular abdominal cavity. Organic urinary bladder is out of the abdominal cavity or the peritoneal cavity, and as it's out of the peritoneal cavity, there's less chance of movement and friction. Urinary bladder, there's only adventitia covering it, not serosa. As you know, the urinary bladder is lined by the transitional epithelium. You know the features of the transitional epithelium. We mentioned that it's stratified there with cells ranging from different shapes. The basal layer, they are cuboidal somewhat, and later on columnar, and later on will change until the surface epithelium becomes the large down-shaped or umbrella-shaped cells, which is mostly multinucleated. As you see here in this slide. This slide urinary bladder with high power, so you can see the epithelium. epithelium, which is the transitional epithelium. Yeah. And thank you.
Merda.